screen.
Because you already started. I'm gonna switch the recording on.
Oh, okay, okay.
That's fine.
Okay, yeah,
Enthusiasm.
yeah. So, may I ask a few questions about the sample shipment to Warwick, please? Because
Yeah.
I'm not quite sure how many milligrams should I send for for three strains of for MRCs to do three strains of bacteria.
Um, I think you could quite happily get away with 10 milligrams.
Really?
Yeah. If you really, really, really were pushing it. Um, if we can do MICs in 96 well plates at something like a 0.2 mil scale, but we can also have the facility to go down to 10 microliters. I mean, essentially, the more you send, the more we can do.
Yeah.
But um, we could basically do what you would probably need us to do with between 10 to 20 milligrams.
Okay. Okay, cool. This time I won't make 50 millimolar. Instead of 50 millimolar, I can just send 10 milligrams, right? Uh,
Yeah, you can do. I mean, as long as we can be certain about the solvent that we're going to use. In other words, as long as we know it's DMSO,
yeah.
that's fine.
Okay. Well, thanks. Thanks for the information. That makes my work much easier. Uh, about these two chemicals. I'm not quite sure whether you guys have the data for the E. coli TOC, E. coli Y type, E. coli K12, and Pseudomonas. I'm not quite sure. Uh,
Okay, right.
I have a.
I'll have to check. I'm away from the lab at the moment. I'm actually in Birmingham. But what I'll do when I get back, in fact, no, I'll drop them an email today.
Yeah, yeah, yeah.
So remind me, we're interested in compounds.
Uh, basically, we've sent the whole table to Jenny, but we were expecting her to check all the data that you guys have for us, but we haven't heard anything back. Uh,
Okay,
so basically,
I'll chase it.
yeah, thank you very much because that would decide how much we're gonna remake. Each of the compounds it will decide the scale, it will decide the purification.
Yeah,
The time we booked for the machine and is about that. So
yeah.
that would be appreciated.
Yeah,
And
so it still remains the point, doesn't it, that IC50 and KIs are we need for the full set. But anything that is promising is MIC.
yeah,
Okay. Anything that I've yet to do, for example, what looks like WYH32, do you want the complete set of ligases, or will Mercy suffice, or what can we do for you?
What have we got on the others, Yuhang? I mean, that's is the consistent set.
Yeah.
I think we only need K12 and the Pseudomonas MIC for these two
No,
strains.
no, but for the IC50, I think.
Oh,
Yeah. Might
oh,
be.
for the data consistency, I think we only need Pseudomonas MUC. Be, uh,
Okay.
because like I don't, do we still have time to do the IC50s for all of these compounds with E. coli MUC? I don't think so, right? Like also we're running out of materials.
Yeah, I mean, a consistent set of data is what's going to be most important.
Yeah.
Paper.
Well, I can prioritize Pseudomonas. That's easy. I mean, we've also got E. coli Mercy in stock, so we could, if you felt it necessary, do that too.
Uh, okay. How much milligrams per sample would be required for E. coli IC50?
Well, okay, I can tell you more or less directly. So, for an IC50, okay, I prepare a serial dilution working in twofold steps back, starting with 10 microliters of 50 millimolar stock. So what does that mean? So that basically means that I can't quite do all the maths in my head, but basically we would have enough um, from, let's say, a mil or even 100 microliters or less.
Okay.
Compound for us is not really limiting, to be honest, in the amounts that you deal with.
Hmm. Yeah, it's the MICs that are the big ones, right?
Yeah.
Um, but it's a good point though. So, yeah, I mean, for the IC50s up there, for all of these compounds, if it's all Pseudomonas, then that's the important thing. Um, I guess I don't know other, how much data we have against other targets biochemically. So, yes, the other Murs and E. coli's, just for the biochemical assays. Because we've only got the two rows there, right, for the in vitro data, not the MIC data, the in vitro data, but do we have data for others that we need to be including in the paper?
Hmm.
I don't know.
Well,
Uh,
well, the question as to whether to include it or not, I think, is probably your decision, but you certainly do have data for the other Pseudomonas ligases for many of these things.
Yeah, so
Going back,
Yuhang, can we dig that up? Because that's important for the paper, isn't it? I mean, it goes to the point about multi-targeting, obviously, but if we've got, if for some of these compounds we've got data across different MERS, then we need to try and make that consistent too. Um, well, I mean, not necessarily across the board, but we need to have, well, if we want to include some of that data, it would be good to have as much of it as we can, I suppose.
yeah.
But yeah. So could you also maybe dig that up and try and add it to the grid here? Because if we do have any obvious holes, now's the time to fill those.
yeah,
But not
sure.
MIC, right? None of that. It's just in vitro.
Yeah, let me dig that out.
Um, and Adrian, for what your data management stuff locally, would you would store when you'd be able to find these data with the Yuhang codes for those molecules, right? That's how
Yes.
you label
Yeah.
it. So
Yeah.
we can do a check between what we have and what you have,
Yeah.
to make sure we're not missing anything for this paper.
Yeah. I mean, perhaps it might be a good idea if today I basically note down everything I've got and just put that into an email so you can see what we do have from
Yeah.
me. And I can also back check that against what the table you've just put in front of me.
Hmm.
And if there are any inconsistencies, like there's something else that's missing, then you can let me know.
Yeah. Yeah. Great. I mean, at this stage, yeah, we're thinking about the in vitro
Yeah.
data. Sorry, the biochemical assay, not the
Yeah.
MIC.
Now, talking of which, I mean, all the data that I've generated has been based upon what appears to be now the justified assumption that they're ATP competitive.
Hmm.
The reason I put it like that is basically based upon Laura's structure,
Hmm.
which shows the compound in the complete enzyme in the ATP binding site. I can, if you feel it necessary, actually corroborate that kinetically. In other words, do a proper set of relationships between kinetic constants and inhibitor concentration, which should agree with ATP competitive inhibition.
Right.
Because thus far, pretty much everything I've done has been based upon that premise. But in actual fact, as far as the entomology is concerned, if I were to be absolutely certain about that point, that's what I would do.
Mm. Yeah. Maybe I mean that would be, I think, very important for a compound,
Yeah,
not for everything.
yeah,
Um, maybe in this last set here, yeah, if it were possible for you to interrogate that, that would be pretty useful for the paper, wouldn't it? Because then we can just say that with certainty.
yeah.
Okay,
Okay.
that would be tremendous. Yeah,
Yeah,
yeah, but not for everything, I don't think, just for
yeah,
one.
yeah,
Uh, I also want to mention that there are two chemicals that we are running out of material. So and the starting material for that is very expensive, 300 pounds per gram. That's why I've been feeling a bit of reluctant to do all of it for the paper. Is that might be the rate determining step for this paper. Uh, yeah, we benefited from the previous antibiotics funding, so we
Hmm.
managed to get it, but we are running out of it. Uh,
Okay, we can talk about that in the lab.
yeah, sure.
I mean, if it's needed for the paper, we've got to do it.
Yeah, I mean, basically, if you don't do it, then
These
Yeah.
um,
we are running out of it.
yeah,
But like I think for us to complete the current table is fine. But
yeah,
if we need more, for example, with other Mur ligases, then we probably run out of materials for sure.
No, but I think the amount of material we need for the biochemical evaluation, the IC50s and stuff versus other Mur ligases is very small. Don't think that makes much difference in terms of the amounts. It's the
no,
MICs that consume the material.
Yeah, for sure. Like for MSCs, I think all the materials, if I consume all the materials, I can get like 15 meg for these two for both either of these two uh eventually. And that'll
Yeah.
be it. That'll be all. And it will barely pass the NMR uh if pure and high res obviously and all the data necessary
Hmm.
and MICs, but it will have nothing left for the
Okay. But I mean, whether we need it, for 87, for example, whether we need it depends on what the IC50 is.
yeah, for sure. Yeah. 87 has a little bit more starting
Uh-huh.
material, but 88 is definitely running out of basically almost everything. Uh, yeah, so that's the thing. Mm.
okay.
Okay. Great. And were you saying earlier that you shipped these compounds to the crick?
Yeah.
Okay. For the accumulation assay.
Yes, I can show you the whole grid. There you go. Share the screen. Uh, yeah, so this is the grid.
Yeah. All the compounds we were just talking about.
Yeah, all
Mhm.
the compounds we were just talking about. So the first line is, if you guys can see it,
Yeah.
first line is the alcohols. The first line are the alcohols,
Yeah.
and the second line and third line are the amines.
Yeah.
The amines we synthesized. Uh, and the third line, sorry, the fourth line is uh the guanidiniums. So this is a oh sorry, I forgot to mark the ice. This is our version of
Hmm.
the AZ595 AZ595, and this is the racemic version of this guanidinium, and this one correspondingly you can see this is the guanidinium version of the 30 WH30, and this is the guanidinium version of 32, uh, 35, 85, and 86 guanidiniums correspondingly.
Where
Uh,
where does the triazine fit in? WYHTRC3.
oh, what do you mean? Like you mean?
Well, I've not seen it before. We haven't tested it, but
Yeah, I'm about to send it. I'm about to send it.
okay.
Uh, it was a starting material. It was a starting material I made for the next round, but I found it super soluble. So and it's di and it's has super soluble in water. So I think it might be worth trying.
It's related to the CC4 carb project.
Yeah,
Yeah.
and I think it's very interesting starting points. So I just wanna fit it in to the grid.
Okay.
Yeah. And the rest are miscellaneous control compound. This is the bulk version. bulk version of the amine, bulk version of the amine, bulk version of the guanidinium, and racemic version of guanidinium.
Yeah.
The last one is the byproduct we got from the reactions.
Yeah.
Yeah, from the SNR substitution reactions. It's just like solvent substituted version. And the reason I selected these chemicals because we have enough starting material. Our previous postdoc helped with the purchase, so we got more starting material. I didn't pick the 88 and 87 bulk version is because we ran out of materials as we just mentioned. These are more easily obtained. And the racemic version, I synthesized the starting material by myself at gram scale, so we got a lot to make. Yeah, that's everything.
Great. Okay. Hi, Chris.
Oh, I don't know.
Quick update. There still seems to be a blockage in getting the protein samples that Laura has generated of the 3 different Murs into the SGC pipeline. And I'm pushing and pushing, but I can't seem to break through to get these things submitted. So I'll try one more time, but yeah, there shouldn't be a blockage here, so I'm not quite sure what's going on. I think it seems to be some IT problem with getting the proteins in the system, but others have managed it, so I'm not quite sure why it's proving to be difficult. But I'll keep pushing because I know the samples have been made and are ready to go.
Can we physically? Where is that? Physically?
Toronto.
Toronto.
Yeah.
I was just going to offer to send them to you. You can just turn up with them and hand them over. It's not that easy.
Right. Yes, that would be simple, wouldn't it? One moment. I've just got to go into the door.
How are you doing, Yuhang?
Yeah, pretty good. Thanks, Chris. How are you?
Fine. So, what's your state of play work-wise? Where are you at with things? I lose track.
You mean
Careers and contracts?
Oh, I'm planning to stay in the group longer. Yeah, for sure. So I'm currently trying to find the right fellowship to apply. Or if not, then I have to find other ways to get a job. But yeah, ideally, my first priority is to stay connected with Matt's group. That's my first priority. Yeah, because I like working with the Mur Ligases team. It's very nice.
So how much time have you got left, funding wise?
For my registration with UCL, I can stay till December. Sorry, November. Oh, sorry, September. Sorry, September. But my current visa extended to January next year. In the meanwhile, I might find other ways of staying around longer. But yeah, definitely.
Cool. Okay, it's good to know.
So yeah, sorry, I'll pursue the protein submission. And what was the other thing? Oh yeah, it was good to hear that the CC4CAR project is still going despite what's happening in the US. So that's a relief. And they're just getting started, I think probably by the end of the month or something, which is good. There was also this new call from Gates and Wellcome about, new initiatives and in antibacterial drug discovery with a focus on Klebsiella. I saw as a model organism. I reached out to the guys at the UK HSA, the Porton-Down folks who have a specialist in that, and they're interested in doing something on new targets. And I was going to propose that we use the SGC hit finding method to go after some, underexplored targets that might be of interest, and pursue those with some compound purchase and med chem and things like that, and maybe some crystallography at Diamond. But I need to come up with a a 5-page draft of something by I think the end of the month or whatever it is, where they're requesting the kind of stage 1 outline proposals. I'm not quite sure how it's going to work. The implication from the call for proposals online is that they will take a bunch of they'll take in the proposals and then may do a mix and match of those, depending on what they receive. That's the reading between the lines. That's what it seems like, but don't know. I don't know if you were looking at that call with interest.
So I missed the bit about the targets because Mandy came in offering me food and I got distracted about the idea of food. So what were you suggesting with Mark for targets for?
Yeah, yes, that little bit there is currently lacking, but we want to.
Yeah. So what we did, probably the last conversation we had before Lara went to Madrid, was to get Klebsiella Mur Ligases cloned up, not surprisingly, and just see what we have that might work against them.
Yeah. I mean, that will be tremendous, right?
So that's our plan. But the time scale, I think, may be too tight for this round.
Yeah.
Right.
I mean, unless we just put in something as a proof of principle. Adrian, you've been thinking about it, haven't you?
Yeah, I mean, we've got sufficient data with things like E. coli, so the call was directed towards Klebsiella and enteric bacteria. So one approach could be to put down what we do have versus E. coli, using that, for example, as preliminary data to look at Klebsiella, which appears to be what they favoured to do. And we were thinking along the lines of Mur ligases, and we were also thinking along the lines of enzymes like Mur, which in enteric bacteria are allosterically activated by intermediates from the PG pathway, so it's a feed-forward activation. So that would provide an additional binding site that's potentially targetable. That they were my sort of first inclinations about what we might do. But I mean, it's still very much something in consideration. We have 2 weeks. The deadline, I think, is the 25th of March to generate the initial proposal.
Yeah. No. So this is the thing. I mean, what I was interested in was using preliminary data on other proteins, which are now published on bioRxiv for the 2 hit finding methods that the SGC is pioneering. So all of that kind of preliminary data that demonstrate that it works is now, public domain. And the idea would be to say that we want to deploy this against some new targets, where you present something which is just underexplored as a new venture. But that to some extent reduces the need by the time of the deadline to produce chemical matter versus those proteins, because you're saying, well, it's worked on all these other targets, so it's a nice method to use. It does get over the persistent hump that we have in going after novel targets in projects like this, that you would in the past need to run a large screen yourself or base your campaign on a known binder. At the moment, the hit rate for these methods is really pretty good, in the sense that you are, for lots of different proteins, finding commercially available molecules that are binding as a starting point. So suddenly the idea of generating hit matter on a lab scale in the short term becomes possible. There's the call out for the AZ MRC screen at the moment, where you can apply to get a full high throughput screen done at AZ, and it's a very expensive proposition, and there are only a few slots available, whereas this thing you can run 8 proteins a week and get some commercially available hits pretty fast that you can then validate with some other method. So it becomes a case of slotting in interesting targets into that proposal, I think. Yeah, I mean, the Murs are interesting, absolutely. So we can carry this on offline if you're interested. I mean, it's 5 pages, right, which includes description of the tech and description of the team and everything. So it's pretty high level.
Yeah, I mean, Chris, I think we could have a conversation about this and then get back.
Yeah.
Okay.
I think.
Yeah.
Because there might be other things we might be able to add.
Okay, great.
Probably will have to do that just remotely, Adrian, over the next few days, won't we?
Yeah.
There's also a deadline for this failsafe community that's run out of Exeter, is it? Which has a deadline of an initial expression of interest for Friday. So I'm currently doing that. That's a fungal consortium.
That's right. There used to be the MRC fungal group that was in Glasgow that all fell out institutionally and just moved wholesale to Exeter.
Right. Okay. Right.
Yeah.
Okay. Other than that, other than the update for the screening for the samples that have been generated and the new call and Yuhang shipment, I don't have anything else. The idea is that once Yuhang's shipped the last samples, we're just going to spend some time, full-time, writing the paper about these molecules, and we'll slot in any new data to the paper as and when they come in, including the accumulation data from Callum and the grip.
Okay. So I take it there was no interest in actual looking at accumulation of precursors.
So it completely depends on your capacity, Adrian, really. And because we don't want to, you've got your own priority list of things here, and if you think that this is something that you can do without it being really burdensome, then again, I think it would be very interesting for the paper, but I don't want to.
I mean, I'm tempted to say, well, what we could do is stick a pin in it, as it were, and if it comes up as a significant referee's point, then come back.
Yeah,
we have to.
I was wondering, Adrian, this is kind of like a side conversation about Lewis's project. To get him to be able to do something which he could do quite easily and get something out of it.
Yeah. So we've got the fresh batch of compounds coming from UCL, the IC50s for those, essentially.
I think that'd be a good thing to get Lewis to do, Adrian.
Yeah.
So it's a PhD student who we've had working on really, really difficult Mur ligases, the ones that form natural chimeras from rare organisms like Borrelia and chlamydia and bordetella. Actually, it's not Borrelia, bordetella. And they're very tricky to work with. So having something slightly easier for him to do, which is a simple bit of biochemistry, I think would cheer him up no end.
Well, I mean, the synergy would be quite nice because one of these fusions is actually chlamydial MRC fused to DDL. So in chlamydia pneumoniae and other chlamydia, those two enzymes actually are basically part of the same coding sequence, and basically you have a bifunctional protein which catalyzes both. So I mean, one certain spur of what we do would be to take some of Yuhang's compounds and see if they do have any activity in the chlamydial context.
But certainly we could get Lewis just to work on a simpler system, a single Mur ligase as well, potentially as part of that. I think so. Yeah, I think the answer is we'll have a go. I guess what it does is it just helps mode of action, internal mode of action validation a little more as well. So. Let's have a conversation with Lewis, Adrian, about doing that once those compounds come in.
Okay. Laura just joined. Hi, Laura, but I think we're all sort of done at the moment. I think we've been through everything. I was just going to say that I'm still pushing for your compounds to get accepted into the Toronto screen. I guess you had no update on that?
No, I need to send them stuff. I just haven't had the time yet because everything is packed, so I need to unpack all the data.
Right. So are they waiting on you?
I think so. They replied that I send them the data to them, and then they will work it out. So I want to finish that this week, so we can hopefully send the samples.
Okay, great. Because it is an interesting one.
It
Yeah,
is.
just because they can run them side by side. And so we would then be able to see if there were any hits across all three.
Yeah, it is very interesting.
Yeah.
Definitely worth trying it.
Yeah, yeah. Okay, cool, great. I won't push in that case. I'll let you do that separately.
Yeah. Hopefully this week will be sorted.
Okay. All right.
Yeah.
Good stuff.
Well,
Okay. Well,
just a quick
Yeah,
question.
sorry I'm late. We just
no,
didn't
don't
have
worry.
the internet for this afternoon, so I had to come home. But yeah.
No, sorry. Hi, Laura. So just a quick question. About the crystal structure you sent us previously. You said it wasn't modified. It wasn't refined yet. So is that structure okay for modeling,
Yes,
or
it is.
uh,
So you are talking about modeling the active side, right?
yeah.
Yeah, so that section should be ready. The rest of them or the protein, I wouldn't do anything with it. It is refined. It's not fully refined, but it is usable.
Oh, it's usable. Okay.
Yeah, and the active side is refined. So
Oh, the active. Okay. Thank you. Thank you. That's a good. Yeah. Thank you very much. Yeah.
no problem.
Cool, very good. Lara, we were just having a quick chat about Klebsiella Mur ligases. I know you just settled in and have not yet unpacked. Where did you get to with ordering constructs for those?
For Klebsiella, we already have one, which is C, I believe. I'll need to check my notes, but there's already one. And then the others, I was just trying to match some orders for the tRNA synthetations on that one as well. So yeah, hopefully this week will be done. So we only need to order 2.
Okay.
I will double check the one that we've got, what are the regions and the construct regions, if it's the full length or not. Yeah. But
And they're with you in Madrid rather than here in Warwick.
if they arrive here, I will be sending you a copy.
Okay.
The Klebsiella Mercy, that one should already be in Warwick as well.
The clone and the protein.
No, only the vector for expressing.
Okay, so we could get someone to express that here.
Yeah.
Okay.
Would you be able to get a quick assessment of how well it expresses?
Yeah. I don't know how busy Louis is, but he might be able to do it.
Yeah, I think we need to have a chat with Lewis. Yeah.
And yeah, because I think he was doing as well other expression tests, so I don't know if he can include this one.
Can you send him an email then telling him where it is?
Yeah. It's on the same box, but yeah.
Yeah, good.
I put everything in the same box. Yes, I will. I have got a picture of the box as well, so I can send it to him.
Good. And I'll try and catch him around now about doing that.
Yep.
You don't have anyone at Warwick who does deep mutational scanning, do you? Of trying to figure out fragility of protein targets, stuff like that?
I don't know, maybe when we had a synthetic biology group, that'd be the type of thing they might have done, but disappeared.
Yeah. Okay.
Is that something you could do in silico nowadays?
I don't know. It's, I mean, possibly. I'm just not sure how valuable it is. As a way of because you really need an assay, you know.
Yeah.
So either it's a biochemical one or more likely a sort of phenotypic one. And seems like, you know, just because you're measuring the death of something, it seems like it's a decent assay would be did it phenotypically. But you know, just instead of just thinking about essentiality, also thinking about mutability as well. This would be interesting to shortlist targets.
Yeah.
Sounds like a lot of experimental work.
Yeah, I think it is, and I don't. To me, it does. We had it done on a viral target, and we got the results and used that for planning. But I don't know how much experimental effort it was.
Hmm. Yeah.
This wasn't a helicase by any chance.
It was the one that we were looking at was a dengue RDRP, sorry, yeah, RNA dependent RNA polymerase.
Yeah.
Just trying to identify binding sites on a protein that we should be targeting.
Yeah.
All right. Well, okay. If there's nothing else, that's fine. It's more of a status update, really.
Mhm.
Yeah.
Yes.
Very good.
Okay, let's be in touch offline about the Klebs thing if we want to go ahead and do something, but let's just
Yeah,
send some email.
we'll press on with trying to get some. In 2 weeks, there's no time. It'll take 2 weeks to actually get the thing expressed, and then
Yeah. Yeah.
So, that's because I couldn't join the, and you're talking about the Belinda and Gates grant.
Yeah.
Yeah.
I couldn't log in into the video, the webinar. But do you require any preliminary data or not?
Well, I assume it always helps.
Yeah.
And as I was just I was just saying that the preliminary data in the things I'm interested in are showing that the screening method that we are using across the SGC works against novel protein targets. And that's the preliminary data, not against anything specific to do with Klebs, but that it is functioning well now and is new, and has a machine learning element to it as well. And then, the targets, we would need to provide something realistic. And for some of the kind of cultures that could be tested on, then the group at Portendown is very interested in doing that work. So, against the nasties. So and against different strains, I guess, and they have a lot of expertise there. So that's a good team. Yeah.
I've spoken quite a lot with Mark. So Mark and his UKHX team are very much involved in what we're trying to do with UK and Japan and hopefully UK, India as well. Kind of lead the program. So we've been talking about trying to share microbiology resources and things when they get overly busy or we're overly busy, and exchange staff between the two. So we're trying to building kind of much closer relationship with them. So it would be definitely good to do something with them on this as well.
Okay.
So you're just thinking of using that platform in Toronto, is that correct?
Yeah, I mean the ultimate aim is to completely reconstitute it in UCL, but we don't have it yet.
What's your timeline on that, do you think?
Well, so yeah, it's more you need a person, and yeah, I'm nearly there with a fully fledged business case for this, but we don't have a person yet. So at the moment we are working with Toronto on the screens.
What type of person do you need?
A mass spec academic. Yeah, someone who knows what they're doing with a mass spec. The kit is not particularly onerous, but you need to have someone who knows what they're doing, and yeah, and who's interested in that area. We have lots of mass spec at UCL, but not of this kind.
So you're looking for academic appointment, academic stream research.
Yeah, that's right. Yeah.
Cool. Yeah, we have lots of mass spec as well. Yeah, but you have to pick and choose who you go to depending on what your question is. Yeah. Okay.
So what are you expecting to have for the grant then?
Don't know. Why don't I'll send the file out, which is not much to speak of, the file after this meeting around to the three of you, and you can see where we're up to. But we are needing another couple of weeks. Let's put it that way.
Yeah. No, that's fine. But at least just to see more or less, and yeah.
Adrian, with your Mur I think if you remember correctly, that's still an opportunity to tag on for a multi-targeting, isn't it?
Yeah, because of the fact it's allosterically activated by UDP Managala. So if you target Mur-D, if you target Mur-C, you would also target Mur-I as well. because you would starve the pathway of UDP-Merlec ALA both ways. And the impact on Mur-I would also negatively impact Mur-D because you're depriving the molecule, the protein of D-glutamate. And it turns out that that allosteric site is found basically in Salmonella, in Enterobacter, in Escherichia, in Klebsiella. In other words, that particular niche set of enteric bacteria, which is why it sort of came to mind.
So from a perspective of, I mean, kind of from a philosophical perspective, say, well, we're going to be multi-targeting, but we're not going to be developing a broad spectrum antimicrobial. That's kind of interesting, possibly.
Yeah, I mean, like I say, it would be a peculiarly enteric drug based upon that premise.
Yeah, I mean, that just sounds an interesting proposition, possibly for someone reviewing it. Right.
Yeah.
Given that they're wanting to focus on Klebsiella.
Yeah. And also, I mean, if I read the call right, they were after. Well, they were out of full proposals for the full amount they were offering, but they were also they did make a statement about for more, how can I put it, more adventurous projects with a higher risk of failure. They were quite willing to put smaller grant funding towards those type of projects.
We can certainly offer them high risk of failure.
Maybe that's not the type of thing we should be stating we can guarantee, but yes. No.
I don't know. I mean, I think that's where we were thinking of going at with it.
Yeah.
But perhaps with a more traditional route. But you know, if it was something that you were prepared to roll the dice again for, roll in another protein with Mur-I, then we'd be very happy to have a go at that.
Yeah. And Mur-I, from an entomological point of view, is a very tractable protein, and it's an essential target in its own right in enteric bacteria. I mean, the only other word of caution is that if you extend beyond that, say you're looking at things like S. pneumoniae and other gram-positives, there are other proteins out there which can fulfill the same function. But within the enteric sphere, then no, it's an essential protein.
I suppose it's slightly different from the Mur ligase program. You're not multi-targeting Mur ligases specifically, but you're picking on Mur-C, which you could argue is the most tractable of them.
Yeah.
And then you know, doubling down with the Mur-I. Yeah.
I mean, it always has been, hasn't it? Because I mean, when we first started out looking at this years and years ago, you know, the point was that the accumulation of UDP-Mernac, if you inhibit Mur-C, is a feedback inhibitor of Mur-A. And because the product of Mur-C, UDP-Mernac-ALA, is an activator of Mur-I, then shutting down Mur-C in its own right might be well be antimicrobial, but also you have the other two impacts, which would also contribute to its ability to act as a good antimicrobial target.
So if you're reducing if you're intrinsically reducing, I don't know, time scale, I don't know, the half-life of Mur-I, but you could argue that you're not actually, you're going to be decreasing the target, the quantity of the target of the second target as well.
Yeah. And also, while we're at it, I mean, thinking laterally, that with Mur-I, because of its mechanism, so it's a racemase, but it's a racemase that uses two thiols to exchange the position of the proton going from L to D-glutamate. And that makes its active site quite susceptible to chemical covalent modification. So that opens up, if you like, another area of investigation, looking at potential covalent inhibitors as well. Again, thinking laterally.
So you could have something that kind of had two modes of action. One, it bound to that substrate binding site for both C and I, and then in I, there was the opportunity to bind covalently that you wouldn't have in Mur-C, but you would have in Mur-I.
Yeah. Well, we say that, but then coming back to, if you all remember the atomized compounds, and there was a series in those atomized compounds which basically the common motif was a thiol linked to an aromatic nucleus. And there was a question which we haven't actually properly addressed yet as to whether they actually acted in a covalent manner, because there was a possibility. That is something that we could look at in quite short order. I mean, if we were sort of conjuring up a project which was to look at developing covalent inhibitors that targeted specifically the Mur ligase pathway and the amino acid pathways that feed into it, it would be an interesting thing to do. As they say, just saying.
So we need to do some mass spec.
Yeah. But that type of mass spec is readily available at Warwick, both here and also elsewhere within SLS and beyond.
Okay. Matt, we're going to explore that a bit further as an option.
Right.
Yeah.
I don't know whether that's something that resonates with you as well, or whether you think of something different.
I think, well, I mean, you know, ultimately it's the discovery and the chemistry that is the bit that's of interest. The novelty for this would be to go after things that are obviously not well explored, and finding starting points is currently going very well. So whatever the targets are, I'm interested in bringing that to it. We also have a pretty good working relationship with the diamond guys who can do a lot of crystallography, and some compound, some hit expansion quickly through methods that they have. So anything of that kind is of interest. We just have to nominate some targets that are strategically useful. Yeah.
So in the selection then, what premium has to be placed upon whether they're, for example, biochemically and enzymologically tractable? I think what you're getting at is if you have a method which basically you're using to select targets, then that pipeline, part of that pipeline will be developing the enzymology to interrogate them.
No, it would be biophysical, initially at least. So it's not an enzymatic assay. So you would check binding through some combination of SPR, thermal shift, NMR, those things.
Yeah.
The biochemical assay would come subsequently.
Yeah.
In the case of the Murs, I mean, there is, you know, you already have worked out a lot of that. But in the case of other novel targets, particularly where you don't know what they're doing, then you wouldn't know what enzymatic assay to generate. And in some cases, the protein may be just, you know, structural.
Yeah.
So yes, it's about binding first, and then, yeah.
Okay.
And I mean, the whole pathway can be finding something which binds well, and then trying to optimize it using a phenotypic assay in parallel, and that, you know, at least temporarily gets around the need for a biochemical assay at all.
Yeah.
At least in the short term.
Cool. Okay, so we'll go and mull over Mur-C, Mur-I, Adrian, Lara.
Yeah.
And, see what we get, and just keep you updated, Matt, as to whether we go with that or not.
Sounds good.
Yeah. Cool.
All right.
Always need chemistry.
Yeah.
Yeah. And if you can send another one, whatever you've got, that would be useful.
Yeah.
Maybe it inspires more targets to suggest.
Very good.
Okay, nice to see everybody.
Hey, you too. Okay. Okay. Larry, are you free for a few minutes after this, or
Yes.
do you want to just give me a call?
Yeah.
And Adrian, we'll have a quick chat, catch up.
Yeah, that's fine.
All right. Okay.
Okay.
Matt.
All right.
Yeah.
See you.
Bye.
Hello.
Bye for
See you
now.
later.
Bye.
Bye.